Hello and welcome to another episode of Nanoparticles versus the Corona Monster. I'm your host, Michael. In today's show, we find out if colloidal copper suspensions are an effective surface treatment against the spread of microbes. Now, ultimately, we want to do these tests on fabrics, textiles, the stuff that PPE is actually made out of. But in order to do that, I'm going to first have to send Jeff Bezos some more money. So in the meantime, I just recreated some of the experiments that were in the papers I read. This just demonstrates whether mixing the colloidal copper suspension in with a growth medium in a petri dish affects the bacterial growth in any way. So since the equipment for the real test is already on order, I didn't really bother filming a video for this. I just made kind of a PowerPoint presentation with a little bit of narration over the top. And as soon as that stuff arrives, I will put out a proper video. Bye! Okie dokie, so my evil plan was to culture bacterial colonies on prepared media plates that were treated with colloidal copper and observe the growth. To make it legit, I wanted to use at least like basic standard practices like the aseptic technique and the don't spill beer all over everything procedure. But since my microbiology skills were about the same as your average 8th grader, I had to watch some YouTube videos first and read a few articles before assembling the equipment for my biosafety level 1 lab. As you can see, I spared no expense on the incubator though. I ordered a live colony of E. Coli. This is the K12 strain, which is commonly used for these baseline antimicrobial tests. Plan A was to dilute the stock copper suspensions down to varying concentrations, spray apply them to the growth media in sections, inoculate the plates with a lawn culture, incubate them, and observe the results. Of course, this was a massive fail. I had no idea really what I was looking at. Plus, I realized I probably spread the copper particles all over the place by lawn culturing with a swab. So, I took a nap and moved on to Plan B. The next plan was to simply rerun the same experiments found in the published literature. This consisted of pouring my own plates with growth media containing varying concentrations of colloidal copper. This time I also streak cultured the plates to minimize spreading the particles while inoculating the plates. Also, I had a little pep talk with the E. Coli beforehand. Each batch consisted of 3 plates, a control, a blank, and then the test plate. The control plate contained growth media only. The test plate contained growth media with a known concentration of copper particles from one of the colloidal copper hydrosols I synthesized in a previous video. The blank plate contained an equal proportion of growth media and a blank sol. Blank meaning the hydrosol was synthesized using the same procedure as the test sol, but substituting the copper salt with distilled water. This controlled for one of the other reagents used in the synthesis affecting the bacterial growth instead of just the copper. The plates were then incubated for 48 hours and periodically observed. In each case, the results were the same. Both the control and blank plates clearly showed streaking, indicating bacterial growth, while the test plate, which contained the copper nanoparticles, showed no streaking and remained clear. This would suggest that the copper nanoparticles, mixed in with the growth media, effectively inhibited the growth of bacteria over a 48-hour period. And I'll call that a success. Unfortunately, all attempts at contacting housekeeping have failed. So until next time, na zdraví and stay safe. See you soon.